sharing our responsibility, especially for the youth.
Pumunta sa farm, naka-PPE. Ang aming ibinabahagi ay yung tamang pamamaraan tungkol sa wastong paggamit ng PPE.
Ayan, no? Good morning everyone. Welcome po sa another uh, ed- edition of our Alam Mo Ba webinar series. Ito po yung ating pang-5. Okay, so if you have attended some of them, ito yung pang-5th or last for this series. Then for today's topic, we're going to talk about herbicide mode of action and herbicide resistance management. So, madami tayong matututunan, marami tayong experts na online. And of course, I'm your moderator. Ako nga po ulit si Edward Rafael Limon, working for Biocrop Science, and of course also a proud member of the Herbicide Resistance Action Committee Philippines ng ating of course ng ating organization CropLife Philippines. So thank you all for joining po. And syempre, I'd like to welcome yung ating participants. So ito, quickly lang check ko lang yung participants list natin. Bigyan natin. Attendees. So madami. Of course, from the different companies. Nandyan po from the, of course, from FPA, from CropLife, from iba-ibang crop protection companies. Ito, I think marami dito students. Thank you. May mga farmers po tayo, may mga dealers, distributors. Ito, ang dami. 216. So welcome po to everyone. Maayong buntag. Good morning. And FPA 12, South Cotabato. Good morning po, Ma'am Mona Lisa. So thank you all for joining. Thank you all for taking time to join yung event. Yan, so maya-maya, palagay po yung inyong name, and then of course some of your affiliations para ma-recognize po namin kayo in your organization or yung school nyo. Pwede rin po, okay? So of course, welcome po sa ating session, and if you have questions, just type them in. Ang ganda po nung lineup natin ngayon. Magaling po yung ating speaker, and then yung panelist po natin is andyan din to support, and of course yung CropLife HRAC team nandyan po. Okay, Batad Green Farmers Association, Sammev Sumagaysay, good morning po sa inyong lahat. Mayong buntag, good morning. Maupay na aga. So yan, okay. So of course today, syempre maraming salamat kasi we are being graced, of course. Nandito po si Sir Eric De Vinagracia, ang ating FPA Deputy Executive Director for Pesticides. So of course to welcome us and to give us his message para po sa ating lahat regarding this webinar po. Sir, good morning po.
Good morning. Good morning, sir. Good morning sa lahat ng participants. I think it's now 226. So again, congratulations advance sa HRAC, sa CropLife for this series 5, the last series for this sessions. And for all the participants, congratulations, lalo na yung nakasunod talaga, no, from series 1 to 5. This is, ito ay napakahalaga sa mga bagay na to, lalo na sa ating pagsasaka. Marami tayong natutunan, tulad ng sabi ng ating moderator, marami tayong matutunan dito. Kaya ini-encourage lahat na magtanong, no, sa mga bagay or mga topic na medyo pag hindi niyo may konting hindi naintindihan, itanong niyo po ito. At again, ang sabi ko nga last session, yung ating pagsasaka ngayon, no, with all the trends, yung global crisis, food crisis, napaka-importante, lalo na sa atin sa Pilipinas, yung ating internal, no, na food production. At sabi ko nga, usually we cannot control the price of our products, no, but we can control the production. Ibig sabihin, kaya nating makontrol no, paano natin padamihin yon sa pagsasaka. At isa na rito yung pagmamanage ng mga herbicides, no, ang mga damo, kumbaga, ma-manage natin. Dahil pag ma-manage natin yan sa ating pagsasaka, napakalaking tulong or napakalaking increase yan sa production ng ating pagsasaka. So in increasing the production, then we will increase the food supply in our country, which is now very critical in these days, no. Alam natin dahil sa kakulangan ang presyo ay nagtataasan, di ba? So pag napataas natin ang produksyon sa pamamagitan ng mga scientifico at yung mga makabagong pamamaraan sa pagkontrol ng mga damo, no, at lahat na sa pagsasaka, ay ito ay malaking tulong. Kaya again, congratulations sa inyong lahat. At just ask, no, pagtanong lang kayo ng tanong kung ano ang dapat ninyong malaman dito. So again, thank you very much and mayong buntag sa tanan, Luzon, Visayas, Mindanao. Magandang umaga po. Good morning po sa lahat. Thank you, sir.
Thank you, Sir Eric. Ganda ng message, Sir Eric. No? We make sure na yung production natin of food is sufficient sa Pilipinas. Ganda, no? Tama yung sabi ni Sir Eric, no, na talagang hindi natin minsan talaga nakokontrol yung presyo, pero at the very least yung production natin, and of course, very related dun sa herbicide management or pag-manage ng damo, we can improve yung ating production. So again, Sir Eric, thank you very much po for always being supportive sa activities po ng Crop Life, especially sa always po giving us yung magandang messages para sa ating mga farmers, students, dealers, distributors, and the different stakeholders po. So thank you very much po, Sir Eric. And then of course, before we move on sa ating programa, i-recognize po natin ang ating esteemed panelists. Ito po yung mga laging sumusuporta, and of course, ito po yung laging mga ito po yung other speakers po natin ng mga nakaraang session. So you might recognize their names. Ang inisip ko kanina dito, ano ba ang tawag sa kanila? Expert? Or minsan, export? Kasi kumbaga internationally known. Of course, itong ating mga panelists. So isa-isahin ko, si Sir Dindo King Donaire, si Dr. Hill Magsino, si Dr. Feli Salas Perez, and then of course si Dr. Nina Gracel Dimaano. So yan po yung minsan pag may tanong po kayo sa chat box, some of them will answer. And then of course yung iba pong questions niyo we'll try to answer live po dito sa ating webinar. So of course to have a glimpse, so tignan po natin kung ano yung topic natin for today. Nood po tayo ng quick video regarding ano ba yung herbicide resistance. So let's see, panoorin natin yung video na to.
Parami ba ng parami ang mga tumutubong damo sa inyong sakahan? Umaabot na ba ito sa punto na tila hindi na namamatay ang mga damo kahit ilang beses ispreyan ng herbicide? Baka herbicide resistance na yan. Ano-ano nga bang dahilan at bakit dumadami ang mga damong hindi na kayang patayin ng mga nakasanayan na laging ginagamit at dating mabisang herbicide laban sa katulad ding damo? At mas importanteng tanong, kaya pa ba itong mapigilan at masolusyonan? Hayaan ninyong ituro namin ang mga paraan kung paano maiiwasan at masusolusyonan ang ganitong problema sa mga sakahan. Sa pamamagitan ng Herbicide Resistance Management.
Magandang araw sa inyong lahat mula sa Crop Life Philippines. Ang damo ay isa sa pangunahing suliranin ng ating mga magsasaka sa kanilang taniman. Kapag hindi lubusang nasugpo o nakontrol, ito ay magdudulot ng pagbaba ng ani at kita hanggang 35%. Kaya naman, mahalaga ang ginagampan ng papel ng mga herbisidyo o herbicides upang epektibong makontrol ang damo sa ating mga taniman. Gayunpaman, kapag hindi wasto ang paggamit sa mga herbisidyong ito ay magdudulot ng herbicide resistance. Dahil po dito, ang Crop Life Philippines ay gumawa ng isang video tutorial upang lubusang maipaliwanag ang herbicide resistance management.
Ano nga bang herbicide resistance at paano ito nag-uumpisa? Ang herbicide resistance ay ang naipapamanang kakayahan ng damo na mabuhay at hindi tabla ng isang uri ng herbicide na kaya namang pumatay ng ibang kauring damo. Dumadami ang damo may sadyang angking tibay laban sa uri na magkakatulad na herbicide base na rin sa paggamit ng magsasaka dito. Habang patuloy ang paulit-ulit na paggamit ng iisang herbicide sa sakahan, dumadami ang mga damong hindi na tinatabla na tumuunti ang bilang ng mga damong kayang puksain ito. Bakit kaya nagkakaganito? Silipin natin kung ano ba nangyari sa isang sakahan. Sa isang sakahan, normal na tumutubo ang mga damo na may iba't ibang biotypes. o mga katangian na may kinalaman sa pagtubo at pagdami ng mga ito. May dalawang uri ng biotypes. Ang una ay ang damo na resistant. Ito yung mga damong hindi namamatay kahit may sprayan ng herbicide. Mayroon ding mga seeds na galing sa katulad na matitibay o di tinatablang damo na matagal nang nakadeposito sa lupa dahil sadyang naipon na. Ngayon, kung patuloy na ginagamit ang produktong di epektibo, mas patuloy na dadami ang mga seeds ng damong matibay. Paalala, namamana ang resistance o ang kakayahan ng damo na hindi tabla ng herbicide. Ang isa pang uri ng biotypes ay ang susceptible o iyong tinatablan. Ito naman yung mga damong madaling mamatay kapag nalagyan ng kaparehas na herbicide. Sa pangkalahatan, marami at iba-iba pa rin ang uri ng mga damo. Kaya naman ginawa ang labeled rates para masigurong epektibo ang pagpatay sa mas maraming uri ng damo. Sa labeled rates, makikita ang tamang active ingredient pati na rin ang wastong dami ng herbicide na gagamitin sa kada ektarya ng sakahan. Dagdag pa rito, siguraduhin gawin ang pag-spray ayon sa rekomendadong timing. Ibig sabihin, dapat sundin ang labeled rates dahil ito ang inirerekomenda ng mga eksperto. Kapag sobra kasi ang ginamit, maaaring masira ang mga pananim at karagdagang gastos lang ito. Kung kaunti at laging kulang naman ang dosage, hindi makakayang patayin ng herbicide ang damo at mas maraming makakalusot na damong kakayanin ng underdose. Suriin natin kung paano nag-uumpisa at dumadami ang mga damong herbicide resistant sa mga sakahan. Halimbawa, makikita natin na sa unang paggamit, mangilan-ngilan lang ang resistant na damo sa sakahan habang marami ang susceptible. Ipagpalagay natin na hindi sinunod ng magsasaka ang nakasulat na labeled rates dahil hindi pa nila alam ang konsepto ng herbicide resistance. Matapos may sprayan ng herbicide, namatay ang mga susceptible na damo o yung mga tinatablan, habang ang resistant na damo o hindi tinatablan ay nabuhay. Lumaki ang resistant na damo at naglabas ng seeds sa lupa. Sa mga unang pagkakataon, successful ang pagkontrol sa damo. Sa kalaunan, kapansin-pansin na mas dumadami ang bilang ng mga resistant na damo. Sa mga susunod pang paggamit ng parehas na produkto, mapupuno na ng mga seeds na resistant ang lupa. At sa pagkakataong ito, bigo na ang magsasaka sa pagpatay ng mga damo sa kanyang sakahan. Ano-ano nga ba ang mga bagay-bagay na nakakaapekto sa bilis ng pagdami ng mga resistant na damo? Paggamit ng iisang uri ng mode of action herbicide ng tuloy-tuloy. Hindi pagsunod sa nakasulat sa label. Dalas ng paggamit ng herbicidyo. Mechanism of action ng herbicidyo. At biology o katangian ng damo. Paano naman malalaman ang level ng herbicide resistance? Ang tindi ng weed resistance ay nakabase sa weed biology at mekanismo. Sa ilang mga kaso, ang tunay na resistance ay nakikita kapag mayroong mga damong nabubuhay matapos ang aplikasyon ng labeled rate at wastong timing batay sa good agricultural practice. May mga pagkakataon pa nga na ang mga damo ay maaaring mabuhay kahit gaano kataas ang aplikasyon kumpara sa labeled rate. May tatlong uri ng herbicide resistance. Ang single herbicide resistance kung saan ang damo ay resistant lamang sa iisang herbicide. Ang cross herbicide resistance kung saan ang damo ay resistant sa dalawa o mahigit pang uri ng herbicide na may parehong mode of action o paraan ng pagpatay. At ang multiple herbicide resistance kung saan ang damo ay resistant sa dalawa o higit pang herbicide na magkaiba ang mode of action. Base sa pag-aaral, ang mga pinakamahusay na paraan para maiwasan ang herbicide resistance sa mga damo ay itinatag sa konsepto ng diversity o pagkakaiba-iba. Ang diversity ay maaaring makamit sa paggamit ng iba't ibang kombinasyon ng mga paraang nagpapababa ng selection pressure ng isang nakasanayang paraan. Ang herbicide resistance management ay pwedeng proactive o reactive. Ang reactive management ay isang estilo ng pamamahala kung saan ito ay tumutugon lamang sa mga nangyayaring pagbabago sa sakahan. Gagawa lamang ng mga hakbang o taktika kapag kumpirmadong resistant o hindi na tinatablan ng herbicide ang mga damo. Ang proactive management naman ay ang pagpapatupad ng mga taktika sa pagkontrol ng damo bago maging kapansin-pansin ang pagdami ng mga damo na herbicide resistant. Isinasaalang-alang nito ang maaaring mangyari sa hinaharap at pinagpaplanuhan ito bago mangyari. Ito ang ilan sa halimbawa ng mga desisyon na kailangang isaalang-alang. Supply at uri ng binhi. Mga kagamitang kakailanganin. Supply ng pataba at ang gagamiting paraan sa pagkontrol ng damo. Pinaniniwala ang kritikal ang proactive na paraan ng pamamahala ng resistance para sa pangmatagalang pagpapanatili ng mga epektibong herbicides. Tulad na lang sa kaso ng glyphosate na epektibo pa rin hanggang ngayon sa GT corn. Ito ang mga pakinabang ng proactive na pamamahala ng resistance. Una, pinapanatili nito ang potensyal ng magandang ani. Pangalawa, matipid ito kumpara sa mga reactive na pamamaraan. Ikatlo, pinipigilan nito ang mga biglaan at panandaliang pagbabago sa mga nakagawiang paraan ng pagsasaka. Panghuli, pinoprotektahan nito ang pagiging epektibo ng mga herbicides para sa hinaharap. Kung mainam ang proactive na pananaw para masolusyonan ang herbicide resistance, ano-ano nga ba ang mga proactive na estratehiyang pwedeng gamitin? Ilan sa mga subok na taktika ay ang paggamit ng herbicide, mechanical at cultural practice. Sa taktikang herbicide, gumagamit ng iba't ibang uri ng herbicides na may iba-ibang mode of action o paraan ng pagpatay sa mga damo. Bukod sa paggamit ng herbicide ayon sa labeled rates, dapat ay tama rin ang timing at paraan ng pag-apply ng chemical na ito. Sa taktikang mechanical naman, gumagamit ng wastong pagbubungkal at pag-aararo ng lupa, bago, habang at pagkatapos ng taniman para makontrol ang damo. Habang sa taktikang cultural naman, gumagamit ng crop rotation o pagpapalit-palit ng tanim. Kailangan din ng tamang espasyo kada tanim, pati eksaktong petsa ng pagtatanim at paglalagay ng mga pataba. Mga iba't ibang paraan ng pag-a-apply ng herbicide na may iba-ibang mode of action o MOA para pamahalaan ang herbicide resistance. Ano-ano nga ba ang mga ito? Kailangang pagsalit-saliti ng paggamit ng mga herbicide base sa iba't ibang modes of action o MOA. Ang dalas ng pagkakaroon ng mga damong resistant ay babagal kapag gumagamit ng iba't ibang mga herbicide na may iba-ibang mode of action. Pinahahaba rin nito ang pagiging epektibo ng produkto. Sa taktikang paggamit ng herbicidyo naman, ito ang mga kailangang tandaan. Ang tank mixing o paggamit ng mga premix na mga produkto na may iba't ibang mga mode of action at aktibidad sa parehong target ng mga damo ay maaaring maging epektibo. Inaantala nito ang pagsisimula ng herbicide-resistant weeds. Sa proactive na pamamahala ng herbicide resistance, importante na sundin ang GAP o Good Agricultural Practices, pati ang paggamit ng full labeled rates. Ang labeled rate ay ang rate o hanay ng mga rates na rehistrado ng Fertilizer and Pesticide Authority. Ito ang rates na epektibo sa pagkontrol ng herbicide resistance, habang underdose rate ay kulang at hindi magbibigay ng epektibong kontrol. Hindi rin ito magkakaloob ng tuloy-tuloy na kontrol sa isang malawak na hanay ng mga kondisyon. Sa huli, ang palagiang exposure sa mababang rate ng herbicide ay humahantong sa ebolusyon ng populasyon na herbicide resistant. Ilan pang mga paalala tungkol sa MOA o mode of action. Kailangang kilalanin at intindihin ng mga growers at extension workers kung aling mga produkto ang maaaring isalit-salit ng paggamit ayon sa mode of action. Habang ang mga kumpanya sa kabilang banda ay dapat naman magbigay ng technical brochure na tumutukoy sa MOA. Mayroon ding mode of action classification ang HRAC o ang Herbicide Resistance Action Network na maaaring pagbasehan. Tumungo naman tayo sa mga mechanical tactics na may iba't ibang mga techniques din tulad ng pre-plant tillage, strip or zone tillage, in-crop cultivation, post-harvest mowing, tillage, manual weeding, hand rugging, hoeing, at weed seed capture. Paalala, kailangan ding panatilihing malinis ang mga equipment at gamit para mapabagal ang pagdami ng mga di tinatablang damo at seeds. Ang proactive management ay nangangailangan din ng mga cultural tactics. Mayroon ding mga agronomic practices tulad ng pagpili ng mga hybrids o iba't ibang mga pagkakaiba sa mga oras ng pagtatanim, fertilizer management, row spacing at mga populasyon ng halaman depende sa hybrid variety, seedbed preparation at mga teknik sa pag-aani. Ang lahat ng mga ito ay maaaring makaimpluwensya sa growth cycle ng mga species at maaari makapagbigay ng kalamangan sa pananim. Halimbawa, ang narrow crop row spacings ay kayang mabilis na mabigyan ng lilim ang mga sensitibong mga species ng weed. Habang mas matagal na panahon ng kontrol ng damo naman, ang karaniwang kinakailangan para sa mas malawak na row spacings. Ang pangangasiwa ng mga damo sa mga borders bago ang pamumulaklak nito ay mahalaga rin upang maiwasan ang pollen movement sa pagitan ng mga resistant at susceptible na mga pananim. Ang pag-iwas sa paglipat ng mga seeds mula sa field patungo sa katabing field sa pamamagitan ng pag-alis ng mga kagamitan bago ito ilipat ay maaari makapagpabakalat ng herbicide resistance. Sa pangkalahatan, ang mga nakagawiang kultura ay may malaking impluwensya sa komposisyon at paglago ng mga damo. Ang mga ito ay nakaka-impluwensya rin sa dami ng weeds at pagkalat ng mga herbicide-resistant weeds. Sana ay marami kayong natutunan sa panonood ng video na ito. Tandaan, pag-aralan at sundin ang mga estratehiyang inyong napanood para maayos na mapamahalaan ang herbicide resistance. Maraming salamat at mabuhay ang mga magsasaka.
Galing, ganda-ganda ng video. So sabi ko nga, sana kung mga estudyante ako may videos na ganito, very helpful. Of course, hindi lang educational, entertaining pa. So in case po, for our all participants, if you want to watch more explainer videos ng hindi lang po ng palay, hindi lang po ng mais, hindi lang po ng iba-ibang crops, crop protection products, stewardship advocacy, and other related topics po regarding sa Crop Life, please po subscribe din sa ating Crop Life YouTube channel. Yan, YouTube channel. Subscribe. Hindi pala nakikita. Nakasubscribe ako dyan sa ating YouTube channel. Type nyo lang po, Crop Life Philippines. Ayan, click nyo lang po yung subscribe. Click yung subscribe para ma-notify kayo kung may bagong videos. Ang dami-dami nyo pong matututunan dyan. And of course, huwag nyo pong kakalimutan, meron din po tayong Facebook page sa ating Crop Life. Dito maririnig niyo po yung ating podcast, subscribe to our newsletter, and of course, visit our website for more helpful resources. So ayan po, naka-flash sa screen, Facebook at Crop Life Philippines. Follow niyo po yan, like niyo po yan, madami pong updates. YouTube channel natin, Crop Life Philippines, ang ating pong website, www.croplife.org, and of course, ang podcast ng Makabagong Magsasaka. So yan, syempre, ano nyo yan, sundan nyo lahat yan. Maganda, marami po kayong matututunan. Well prepared po yung mga videos. Marami ring statements, of course, from the different partners ng ating pong Crop Life Philippines. Okay? So, yan. So, ano nyo yan? Ngayon pa lang, may tip ako sa inyo. Atin lang. Kunti lang naman tayo eh. Subscribe kayo tsaka follow kayo kasi may pag-games mamaya. Para manalo kayo, kailangan subscribe and follow. Okay? Okay, moving on with the program. I'd like to call on, of course, yung representative natin sa Asia Regional Herbicide Resistance Action Committee. So ito, kumbaga sa term natin sa Pilipino, mga imported to. Okay? Ang ating core team member, of course, from colleague ko din to, from Bayer Crop Science, co-representative din. Asia Regional HRAC, of course. And of course, supporting ating National HRAC Committee din, si Sir Ramesis Fulgencio o si Sir Ram Fulgencio. Sir Ram, good morning. Good morning, Sir Ram.
Yeah, good morning, Edward. Can you hear me? Okay ba?
Yes, Sir Ram. Okay naman.
Okay. So yeah, again, thank you, Edward, sa very kind introduction. So, I mean, good morning to all participating online. So, magandang umaga, maayong buntag, maupay nga aga sa inyong lahat. So, maybe, I think, Edward, there are also may mga international, I think, participants din na in this call. So, please allow me to give my remarks in English. Okay? Sana okay lang yun. So, okay. I think it is the 5th of the series, but already on behalf of Asia HRAC, I would like to thank Crop Life and HRAC Philippines for once again inviting HRAC regional team to another valuable session in your lecture series. I remember I've already joined, I think, 2 in rice and also in corn. And now, basically on this very important subject of mode of action and herbicide resistance management. So there has been few discoveries actually, kung alam nyo na, in the last 30 years, and we would not expect much of herbicide discoveries, I would say, in the future. Hence, one of the mission of HRAC basically is to prolong the effectiveness of the herbicides. So meaning, knowledge or appreciation of herbicide mode of action is very critical part in the process of developing any herbicide resistance management program. So meaning understanding how herbicides are absorbed into the plant, translocated in the plant, metabolism of the herbicide, and physiological plant response. This would be the topic involved, I think, in the mode of action later. In terms of chemical control component integrated with management, we have already always preached of rotating and mixing different herbicide mode of action, which has already been proven to delay and manage resistance buildup, among other methods. Pero dapat natin gawin to in a more sustainable manner. And I think understanding or different herbicide mode of action, like I mentioned, would help in combining the correct mode of actions. So I believe our distinguished speaker today, Dr. Burgos, would elaborate on this further later. Okay, so furthermore, it is HRAC advocacy. As you know, in previous, we introduced advocacy of HRAC to drive the mandatory mode of action labeling with regulatory authorities and all stakeholders. So hence today's topic is really timely, to understand why we need to do this. So I see it was mentioned 200 plus participants in today's call. So again, a big congratulations to CropLife and HRAC Philippines. I'm sure this would be another successful webinar. So good morning again and thank you.
Thank you, Sir Ram. So Sir Ram, yung nabanggit mo, 250 sa Zoom, I'm sure a lot more pa, baka doble pa or triple pa sa ating Facebook. So yung mga nanonood ng live stream. So thank you, Sir Ram, for explaining kung paano ba natin nabibridge yung ating science. Then of course, going into practical or syempre sa ating yung pinaka-affected sa ating mga farmers. So Sir Ram, thank you very much and I'm sure, I think, aalis si Sir Ram, mag-feel din yan regarding din sa herbicide resistance control. So I think he's leaving then also. So thank you, Sir Ram, for the time. And then of course, before I call on sa ating next speaker, meron akong good news for everyone. Except me yata, hindi yata ako pwede sumali. You can join and win cash prizes. Okay, cash prizes during the trivia game. Ito, binulong ko sa inyo kanina to. To qualify, participants should be subscribed dun sa CropLife's YouTube channel. Very easy lang naman. CropLife, type nilang CropLife Philippines, subscribe lang kayo dyan. Okay? And you should like and follow yung CropLife's Facebook page. Okay? At CropLife Philippines, like and follow. And then of course, syempre ulitin ko lang, kung nakasubscribe na kayo, sayang din ang ating premyo na pwedeng makuha today. Ayan. Ayan. Thank you, Sir Bo. Ayan, subscribe sa CropLife Philippines YouTube channel. Subscribe kayo, click nyo lang yan. Follow, like and follow our Facebook page. And then sa Q&A natin, may isang person na makakasagot pag may premyo, may mga iba-ibang winner from Zoom. May 3 winners from Zoom, meron ding 3 winners from Facebook. And of course, ino-notify naman po namin kayo pag kayo po ay nanalo, and then prizes will be sent through GCash. So GCash, ang daming pwedeng gamitin yan. Pangbayad ng bills, pangbili ng pagkain. I think yung ibang agri-supply, matanggap na ng GCash. So daming ano. So ano yan, good as cash na rin yung GCash na yan. So yan. So sana sumali kayo. Ang daming winners. So 6 na winners natin for today. I mean, per question yata yan, 6. Pero anyway, subscribe po kayo sa ating YouTube channel, sa ating Facebook page. Follow niyo po yan, support po natin. And then yung mga kamag-anak po nating magsasaka, kamag-anak nating dealers, distributors, yung mga classmate po natin sa university, workmates natin, like and subscribe niyo po yan. Of course, to help the channel and the Facebook page grow. Okay? And eto na, papunta na po tayo sa exciting part ng ating webinar for today. Definitely, I'd like to introduce yung ating speaker or ang ating expert for today's webinar. Ito yung profile niya, just looking at it, ano ang feeling ko masasabi ng ating mga participants ay sana all po. Educational background po is of course ng kanyang bachelor degree 1983 sa Visayas State College of Agriculture or VISCA. Iba na po yung pangalan niya ngayon sa Leyte. Philippines Agronomy Soil Fertility, 1994, natapos po niya ang kanyang MS sa University of Arkansas. Hopefully, ma'am, na-pronounce ko na maayos po yung University, Agronomy with Science. And then, of course, yung kanyang PhD, again, also sa esteemed University of Arkansas, Agronomy with Science with Physiology. Marami rin po siyang research activities with physiology, ecology, biology, molecular biology, transcriptomics with emphasis on weed adaptation to herbicide selection, pressure and abiotic stress. Also conducting research on weed management in rice, soybean, vegetable crops. Yan, kasama dyan yung cowpea, cucurbit, sweet potato, and blackberry. Publications, ayan, books and book chapters 10, referred journal articles 135. So sobrang dami. And honor and awards, meron siyang 32. Sige nyo, 32. Sobrang dami. Pero just to name a few, International Education Award, Division of Agriculture, University of Arkansas. So yung 2022, ganyan lang. And of course, this syempre makes us proud. Diba? Talagang our Filipina doctors talagang well representing ng ating country and bringing yung mga ganitong international awards, Fellow with Science Society of America. Okay? Outstanding Educator, Southern With Science Society. Outstanding Alumni ng Visayas State University ng 2013. And then of course, mentor ng first IWSS Larry Pearl Awardee International With Science Congress noong 2008. And ito, ang kagandahan po dito is si Doc Nilda Burgos, of course, ang ating guest speaker, Professor with Physiology and Molecular Biology, University of Arkansas. At mo, binasa ko lahat ng hindi ko pa nabanggit pala yung pangalan niya. Again, ang ating presenter for today, si Dr. Nilda Burgos. Nakausap ko siya sa dami po ng accolades niya, awards, honors, educational background. Talagang during nung quick time namin na nakapag-usap, very, very down to earth po si Ma'am Nilda. And I think, mamay ko kwento sa inyo ni Ma'am Nilda kung ano oras na. Sa kanya again, Ma'am Nilda, thank you very much po for accepting the invite and of course, imparting yung inyong knowledge and experience sa amin po, sa ating pong participants, sa ating pong mga Filipino farmers to of course, really understand more yung ating mode of action and of course, resistance management. So, over to you, Doc Nilda. Good morning.
Good morning. Maraming salamat, Edward Limon. Thank you so much. Good morning, everybody, those who are online and watching this webinar. And magpapasalamat ako sa CropLife Philippines and HRAC Philippines for inviting me to participate in this activity para maka-contribute naman tayo sa pagbahagi ng mga impormasyon. So anyway, hihingi ako ng paumanhin sa mga audience na kung halimbawa yung mga audience na matagal nang wala sa university, matagal nang wala sa classroom, kasi ito kasing topic na herbicide mode of action, ano siya, yung it tends to be masyado siyang technical, so at saka hindi ako prepared na i-translate yung karamihan ng mga terminology. So parang sabi ko, siguro para naman magampanan natin itong topic na ito sa oras na meron tayo, siguro majority nito ay English na lang, okay? So sorry about that, pero I hope na makakuha naman kayo ng mga main points dito sa presentation na ito, okay? So let me start. I-share ko yung screen ko para makita nyo yung presentation. Okay. So I'm here in Fayetteville, Arkansas. Nandito pa ako sa aking office. It's 9:15 in the evening now. So alas 9 pasado na ng gabi. So anyway, simula na tayo, okay? So ngayon, ang topic natin ay itong herbicide resistance. Meron tayong maraming grupo ng herbicides, mga 26. So I asked Janina kung kailangan nating i-focus kasi konti lang ang time, ano? So yung medyo mas may relevance dyan sa inyo. So ang advice niya ay yung i-focus natin doon sa mga mode of action na kung ginagamit sa rice production para kumbaga ang model natin ay, or magiging example natin most of the time ay yung mga herbicide na nagagamit sa rice. And then, so, merong sinasuggest si Janina ng mga mode of action groups. So I listed them here, pero hindi ko na sinali yung HPPD kasi sabi ko baka wala ng time, ano. And then kanina sa presentation ng herbicide resistance management, I'm so glad you did that kasi lahat ng mga detalye na yun is everything that we have to talk about sa resistance management part, and you did an excellent job putting together that video. So I'm so glad that you did that kasi ang ginawa ko lang dito sa aking presentation is really just to outline an overview of the principles kasi I was intending na we can discuss that later during sa Q&A, no, if you have more detailed questions about resistance management. So ito yung link ng ano na yon, yung mode of action map na yon, yung poster na yon, so you can actually get that. You can download it. Yung mga estudyante, kung gusto niyo yung poster na yun, you can download it. So as an overview, yung herbicide groups na ginagamit sa rice globally, okay? So hindi ito lahat na ginagamit sa Pilipinas. So hindi ko alam kung ilan dito ang ginagamit sa Pilipinas, pero globally ganito kadami ang mga herbicides na ginagamit sa rice. So actually, merong mga 13 mode of action groups. Okay, so ano lang, as an aside, in relation to herbicide resistance management. So this is to get us down to earth again, like get us all grounded back to our reality. So isa sa mga principles of resistance management, of course, is diversification. So maalala nyo siguro yan, kung mag-quiz kayo later, baka isa sa mga questions nyo ay ano yung iba-ibang mga mga items for resistance management. So one of those big ones is diversification of herbicide mode of action. Tingnan natin itong rice. So there are 13 modes of action here. Okay? Tapos ang dami-dami mga examples ng herbicide dito. Ngayon, tingnan naman natin dito sa baba, itong nasa red. Ilan ang mga resistant weeds sa rice doon sa website ni Ian Heap, sa Weed Science Society of America, or sa Weed Science or org website. May mga 43. So ang dami, ano, so sabi natin, okay, so ano, paano nangyari yan? How did it happen? Ang dami nating herbicide sa rice, we have this many, meron pa rin maraming resistant weeds. So ngayon maisip natin na isa lang yan. So diversification of herbicide mode of action is just one aspect of resistance management. Marami pang many other aspects na pinapakita doon sa video. Ibig sabihin, kung i-review natin yun at pag-aralan natin yun, marami tayong mga factors na kailangan nating isali doon sa ating resistance management strategies. Okay? So,
Doc Nilda, sorry. Nag-share po kayo ng screen?
oh, yeah, I did.
Parang wala pong nag-show.
Ay, ganun ba?
Ayan po, Doc. Thank you, Doc.
My goodness, sorry. Okay. So ito na yung binasabi ko na ano, herbicide sa rice na 13. Sorry about that. I didn't. Oh, thank you for telling me. Okay. So okay, ngayon ay punta tayo sa first group. So yung sinasabi ko na groups ng herbicides na i-discuss natin ito na ngayon sa ano. Punta tayo dito sa first group, dito sa Group 1 ay tinatawag natin lipid synthesis inhibitors. So kasi ang epekto nito when we inhibit that ACC-ACE enzyme na tinatawag natin, it will result in the disruption of lipid synthesis inhibitors. So bakit mamatay ang mga tanim kung wala na itong lipid synthesis inhibitor pathway? Well, tingnan natin kung ano yung mga lipids. So lipids are fatty acids, and then complex lipids comprising membranes, waxes. Waxes yung mga leaves ng plant, may mga waxes yon. Pigments ng plants. So pigments, ano ito? Chlorophyll, yung lahat ng mga ginagamit sa photosynthesis. Lahat yan. Even yung mga vitamins, vitamin A, D, E, and K. So lahat yan ay composed of some types of lipids. So maraming type ng lipids. Tayo, mga tao, we are composed of lipids. Kung walang lipids, patay tayong lahat. We would never exist. So may lipids na may lipids na useful, may lipids in good amounts, and then of course when they are there in too high amounts, it could be bad. Halimbawa tayo, kung masyadong mataas yung cholesterol natin, it's bad. Pero kung nasa balance yan, it's good. We need it. So the plants are the same, so they need that. So this group 1 herbicides act here. So itong this is the first step of lipid synthesis. This one is production of acetyl-CoA. Ito yung isang ingredient to make lipids. Lipids basically is a chain of carbons. And so then this acetyl-CoA needs to be lengthened. Kailangang pahabain siya. So we need to add more carbon to it. So ngayon, kailangang magiging 3 carbon siya sa next step, magiging 3 carbon siya, magiging malonyl-CoA. Ang enzyme or protein na mag-facilitate sa reaction na ito is ACCase, ang tawag natin, no? Acetyl-CoA carboxylase. Ang reaction na ito ay, sa tingin natin is simple, but it is difficult. So enzymes are proteins that facilitate difficult reactions. So sa class yan ang ituturo sa atin, ano? So ang target sa pathway na ito is to make lipids that are having 16 carbons to 18 carbons, and those are the types of lipids we talked about earlier. So ngayon ito yung ACCase inhibitor. Dito sila, this is where they act. And so what are the herbicides that are inhibiting that particular enzyme, this ACCase enzyme? So meron tayong 3 families of herbicides that act in this group. First one would be what we call aryl oxyphenoxypropanoates. In short, we call them FOPs. Kasi yung mga pangalan nila, they end in FOPs. So easier to call them that way. The second group would be this gray boxes here in the middle. This would be the cyclohexindiones. Ang mga pangalan nila ay mag-end ng dim. So clisodem, cetoxidem, these are just 2 examples. There are several others in here. And so ang tawag sa kanila DEMS. And then ang pinakabago sa group na ito, the newest one, well, it's not new anymore, but relative to the others, the most recent one would be this one, pinoxidine. So ito yun siya dito sa baba. So itong mga herbicide na ito, these are called selective grass herbicides. These are only effective on grasses. It could be annual grasses or perennial grasses. So they are translocated in the plant. They're called lipophilic kasi or oily kasi hindi sila mag-merge sa water, no? Parang oil, so they're not gonna merge with water. So in that sense, they can cross the plant membranes easily. So makakross sila sa waxes ng leaves and sa cell membranes easily kasi they're kind of oil loving, so they can do that, and then they get translocated in the plant via the xylem. So systemic ito. So pag mag-spray ka ng ganito, even if the spray droplets are bigger, or even if the plant is not covered 100% with the spray, maka-translocate yung mga herbicides. So coverage is not as critical compared to herbicides na sinatawag natin contact herbicides na hindi maka-move. Ito sila maka-move. Although hindi natin, we should not abuse this part. We should still strive for proper coverage whenever we spray, para even ang coverage, and then lahat ng plants pareho ang na-receive nilang dose, and then we are not going to expose other plants to low doses na that would allow them to survive and be selected later on for resistance. Okay? So they are slower acting kasi nga systemic siya. It takes a while to inhibit. The lipid synthesis inhibition will take a while to take effect. Pagpunta natin sa field, if we apply these herbicides, we normally see grasses that become kind of reddish, purplish, na ganito, parang mapula, parang purple, ganyan. So because of the action of these herbicides, the plant of the part of the plant that is affected would be the growing parts of the plants. Kasi doon yung plastids and doon yung beginning ng elongation ng mga lipids. So yung growing point ng grasses, these are the ones that would be affected. Okay, so ma-deteriorate yan siya. The broadleaf plants will not be affected by these. So wala din ito sila generally, wala silang soil activity. So itong ganitong herbicide, you have to use this foliar. So apply mo sa weeds na tumutubo na. And then you cannot expect that to control the seeds na hindi pa tumubo. So you'll have to pair this with something else in order na makontrol yung hindi pa tumubo or yung mga damo na tutubo after. Ang one thing that we need to be aware of when we use this grass herbicide is they have a high risk of being antagonized by other herbicides that have activities on broadleaf weeds. So we have to be careful in using this para hindi natin siya basta-basta i-mix. Kasi di ba one of the approaches for resistance management is mixing different herbicides, right? So then when we're mixing with these kinds of herbicides, we have to be careful. We need to check the information on the label kasi nilalagay doon kung compatible ba ito siya to a certain herbicide. Mayroong example ditong ginawa, so halimbawa 2,4-D or Cetoxidem, hindi siya compatible sa itong mga FOP na herbicide na ito. They're not compatible together, 2,4-D and Cetoxidem, for example. So yun lang ang ibig nating we need to remember about that. So at this point, I'd like to review yung overview sa resistance mechanisms kasi nga yung topic natin ay kasali din ang resistance. So may dalawang uri ng resistance mechanism, malaking grupo. So one of these is non-target modifications, meaning target ng herbicides. So halimbawa itong ACCase herbicides, ang target nila ay the enzyme ACCase, right? So yung mga plants na could become resistant to group 1 herbicides or ACCase herbicides, they could either have modifications outside of the target, so outside of the ACCase enzymes, or they could have modifications or mutations within the enzyme. So itong diagram na ito, kinuha ko ito sa publication ni Dr. Delie, is showing that. So different ways the plants can acquire resistance. So itong A, B, C, D, and E, mga non-target. So A, reduced amount of entry into the leaves. So waxes maybe are modified, hindi makapasok ang herbicide or less herbicide can go in. B would be translocation within the plant, so movement within the plant. So that could be facilitated by transporters or something else, and then if it cannot move enough, then the plant will not be killed because there's less herbicide reaching the target. And so on and so forth. Ngayon, ito namang modification sa target is mutation where the herbicide binds. So it's an enzyme. Of course, it binds the substrate to facilitate the reaction, right? So in this case, herbicide would replace the substrate and then bind into the enzyme instead. So yung mga modifications na yon are classified into different ways as well. So my modification na would increase the fit of the herbicide. And so then it would be very resistant, right? And then my modification din na it alters binding and it fits a little bit better pero not as snug, hindi masyadong tight. And then that could give like lower level of resistance. So depending on how the modification kind of increases the fit or kicks out the herbicide, if it kicks out the herbicide, it becomes resistant. If the modification makes the herbicide their fit better, then it becomes more sensitive. So like the other way around, right? So normally this modification, the one that results into resistance is when it kicks out the herbicide because it cannot fit in the binding site anymore. So yun ang ang principle. Ngayon, this ECCA's herbicides, there are a lot of weed species that are resistant to them. In the website at the herbicide resistance survey, there are 50 that are listed as of last year. The majority of reasons for these weeds to become resistant is because of mutations in the ACCAs. And so yung karamihan nito of course ay grasses. So itong mga foxtail, mga lolium, mga ganyan, Johnson grass, meron silang mga mutations in the ACCase enzyme that prevents the herbicide from binding anymore or reduces binding efficiency, and then it makes them resistant to the herbicide. Itong naman, the second group of herbicide is going to be group 2. So itong group 2, tinatawag nating ALS inhibitors. Itong ALS ay another enzyme naman ito. This enzyme is one that facilitates the synthesis of amino acids that have branched side chains. So tingnan natin ito. So ngayon, ito yung pathway ng group 2 that is inhibited by group 2 herbicides. Ito yung pathway ng paggawa ng branched chain amino acids. So this would be isoleucine via this path here that is facilitated by AHAS, and then the other path would be synthesizing valine and leucine using this ALS enzyme. These enzymes are kind of more or less isoforms. They have similar function, facilitating this reaction. And so this inhibitors would actually act here in this place where the ALS enzyme is facilitating the union of two pyruvate molecules to make this intermediate right here. So ang makita natin, if we remember where the ACCase was and where the ALS is, the most effective herbicides are herbicides that actually stop the pathway at the very beginning, because then that would be the most effective way of stopping everything from happening downstream. So this would be like one of the principles of herbicide design, is look for a point where you can stop almost everything. So it would be the most effective target. That's why we have this. So ALS inhibitors. So what are these ALS inhibitors? A lot of them. So in all of the herbicide groups, if you look on that whole poster of HRAC, you will see that ALS inhibitors compose the largest group of herbicides. So there are 6 herbicide families here: imidazolinones, sulfonylureas, these pink ones here, pyrimidinal benzoates, sulfonanylides, triazolinones, and these triazolopyrimidines, 2 types of triazolopyrimidines. So this is good. There's a lot of herbicides that are in this group. Well, on the flip side of that is, so it's a double-edged sword. It's good because we have a lot of options. On the flip side of it is that there are a lot of it offers a lot of selection pressure. So then now we also end up with a lot of resistance problems against this group of herbicides. So a lot of herbicides here are compromised by resistance. So anyway, overall, these herbicides are going to be also systemic, so they can be absorbed root or shoot, and then it takes a while for the plants to die. And then, although the activity actually starts very soon, within 2 hours after treatment, there's already effect on the cells, but the death is slow. So makikita natin in the field would be plants that are generally stunted, they're yellowish, and they're purplish and all that, just totally unhealthy. And some of the stronger ones can actually also cause burning of the leaves. So masusunog yung mga dahon nila, para bagang nasunog, pero talaga namang namatay lang yung mga tissues, no? And so the difference among species, so spectrum na ito, kung ano yung mapatay nila, ano yung susceptible, ano yung hindi, it really depends on the ability of the weed to detoxify the herbicide. So ito yung differential metabolism na sinasabi. itong differential metabolism ay natural, natural defenses sa plants to help them survive something na would poison them. Pero kung titingnan natin, itong mga natural defenses ng weeds, ito din yung nagiging mechanism for resistance to herbicides. Ito din yung nasiselect natin for the weeds to become resistant, if we think about it, eventually. So in the field, ang makikita natin ay ganito. So itong example na ito is imidazolapyr, but it represents the imidazolinone. So makikita nyo yung cotton is very yellow. This is sickle pod, Synoptosifolia, is very similar to peanut, and this is grain sorghum, and then this is rice. So rice, susceptible to imidazolinone, would have this kind of yellowing and all of that. And then the crops that are tolerant to this are the legumes. So ito ay bush bean, halimbawa dito, soybean will also be tolerant. Kita nitong grasses is susceptible. And then another example, so another family, the sulfonylurea, trifloxysulfuron. This one is kind of stronger herbicide compared to imidazoline, that particular example earlier. But this herbicide is used here in North America for cotton. And so soybean is not tolerant to that. So soybean, you'll see it's stunted, it has purple veins. And then here's corn, it's not tolerant to that as well. And even peanut is not tolerant to it. So you will see a lot of yellowing and stunting. So this is kind of the general picture when you look at a lot of the ALS inhibitors will kind of show similar look. And there's triazolopyrimidine here. So this example is flumetulam. It's not used in the Philippines, but it's also a soybean herbicide and corn. But then you'll also see stunting here. So this is morning glory, Ipomea, the weedy relative of sweet potato. It's going to be stunted by this herbicide. And then soybean will be okay. And then grain sorghum is going to be practically killed by the herbicide. It's not dead, but it's very stunted and yellow. So same principle. And then here's pyrethiobac, which is pyrimidinal benzoate in rice. This is going to be similar to bispiribac. And so it's active on Sesbania, and then it's going to yellow the soybean, it's going to yellow the morning glories, and it's going to yellow the sorghum. But this one is a cotton herbicide. And so its relative is used in rice, the bispiribac sodium, for controlling grass. So ngayon, so how do weeds become resistant to this? So can you imagine there are 170 cases of resistance to ALS inhibitors worldwide. So that's a big problem. It doesn't mean na itong group ng herbicides na ito, they are all compromised. There are many herbicides in this group that are very useful that we couldn't afford to lose. And actually, yung pag-manage natin ng resistance, our goal is actually to preserve the herbicides that are still useful in any of these groups because we need all the tools that we can use. So, ang makita natin, later we will see na the majority of the resistance mechanisms to ALS inhibitors is because of mutations, because of changes in the binding sites. So yung close-up ng binding site is kind of a 3D picture of the ALS enzyme. And then this is a sulfonylurea here. So this is like the shape of that molecule, sulfonylurea. And then the binding, the catalytic site of the enzyme forms a pocket, like a cavity pocket. And then the substrates, yung mga pyruvate, papasok sila dito. So then they can be bound here by the amino acids here, and then they can be stabilized so that, you know, the acetolactate can be formed. So it's like forcing them to go together. They don't like each other, but then there's the enzyme that forces them to go together, then they form something. And so, which is a necessary product. So ngayon, pero pag nandito yung herbicide, the herbicide gets in here, and then it binds in the amino acids that are here in this proximity. Then when it's there, then it blocks the path of the substrate to go in. So it excludes the substrate. So meaning it's competitive binding, competes for this space. In similar manner, this imidazolinone herbicide here, it also goes into this, course, similar, the same packet, but their binding region slightly differs. It differs slightly. So you can see a slight difference in the shape here. And so there's also a slight very minor differences in amino acids that are involved in binding each one of these. So we can imagine that if any one of these key amino acids that are holding the herbicides here, if any one of these changes, and then it precludes the herbicide from going in there, or it loosens the binding strength, then it can make the plant resistant, the weed resistant. And so that's actually what's happening. So then we can see here, so here's an example, here's a list of mutations. Alanine mutation at position 122, and this alanine can be replaced with a threonine or a tyrosine. It results in all sorts of resistance. So for example, it results in resistance to imidazolinone, and then low resistance to sulfonylurea, right? And then if it is replaced with tyrosine, then it becomes highly resistant to sulfonylurea and triazolopyrimidine and imidazolinone. So it depends on what's replacing it. Then it changes the resistance profile. So see what we now. So see what we now have. So now when we look at resistance management, we actually would be informed. Could decide better what kind of rotations we can use or replacement of herbicides we can use, even if it is in the same group, if we know the resistance pattern and the mechanism, and then the profile of the weed population. See, so it is important to understand what is making it resistant. Then it would help us in our rotations of herbicides and mixing herbicides. There are mutations that would make the weed either healthier or it will make the weed kind of sick. So meaning that there is mutation that is going to allow the weed to survive the herbicide application, but that mutation makes the enzyme less effective because it couldn't bind its normal substrate very well anymore. Then it becomes sick. So here's an example here. So this is Raphanus, this is what is that, wild radish, and this is a study in Australia. And so this particular mutation here is tryptophan at 574 replaced with leucine amino acid. So that mutation is not very favorable for the weed health, for plant health, but it allows it to survive a little bit. But so, and then here's, in comparison, here's another mutation here, alanine 122 to tyrosine. It makes the plant a lot bigger. So if this is a weed, as this is a weed, wild radish, so you would wish that the mutation would be something like this, because the weed is not healthy. Then you can overcome, you can manage that resistance easily, you know, by proper crop spacing, etc., competitive crop and all of that. It's not very competitive. But if the mutation is this, then it's hard to manage that with cultural management only. It probably should be managed with a total replacement of the herbicide, right? Totally different mode of action, something that would just kill that right off. And so cannot rely on competition to manage that. So there's also that aspect. This picture, this figure here, actually I lifted from Nina's, Dr. Dimano's paper. She wrote a really nice review paper in Pest Management Science, published last year. That is good reading for the students here who are listening. You should read that. That's very informative. And that talks about the role of cytochrome P450s, and that's related to herbicide detoxification, and also related to the situation where today we could have a new herbicide that is just introduced. And then it would turn out that we apply that herbicide into the field and there are already weeds in the rice field that are resistant to it, when we haven't used that herbicide before even. Well, one of the reasons for that is the weed population that is in the field, those have already been selected for resistance to other herbicides. And then if that resistance is because of increased activity of this group of enzymes we call cytochrome P450s, for example, so that means that this population is able to detoxify the herbicide faster. The activity of this cytochrome P450s actually is not totally understood yet. We're just starting to understand this, and probably Nina talked about this. And they can act on multiple herbicides, multiple substrates, right? So it is possible that if the weeds now are resistant because of a particular P450 that detoxifies herbicide, that P450 could potentially also detoxify the new herbicide. Then we are faced with, already, we're already kind of behind because the weight is already advanced in terms of its evolution. And that's just one of the realities that we face today. It's a little bit more difficult now, a little bit more complicated. So tayo, kailangan nga, kaya nga sabi dun sa video, we have to be very careful and we have to pay attention to what we're doing. Kasi hindi na simple ang mga weeds ngayon. So now we go to another group. This one you're probably more familiar with because this is where glyphosate is. This is EPSPS inhibition. So, and the only herbicide in this group is glyphosate. So we have to use that as an example. So ang ano naman ito is still amino acids, but these amino acids now is a different type of amino acid. These are the aromatic amino acids. So these are amino acids where their side chain is a benzene ring, aromatic ring. So it's tryptophan, tyrosine, and phenylalanine. So the reason, here's the pathway here. Where does the enzyme target rest? It's here in this step of making shikimate 3-phosphate into enol pyruvate, shikimate phosphate. And so that's facilitated by this enzyme, EPSPS. We just call it that kasi mataas yung kanyang, mahaba yung kanyang pangalan, ano? So EPSPS. This is a crucial step right here. If we stop the reaction, the pathway here, none of these things here happen downstream. But then if we look at the products downstream, can you imagine the diversity of these products and all the function of the plants that are facilitated or made possible by these products? So IAA, what is that? That's a basic hormone in the plant. So that is facilitated by, that is a product coming out of tryptophan, right? And so even if we just look at that, if we stop that, the plant will die. So that's why glyphosate is so effective, because it is acting on a place where if we stop that, it removes all these, and so no plant can survive that. And that's why glyphosate is non-selective. So this is one of the most effective non-selective herbicide we have. And so this glyphosate has activity in this step. So it inhibits this particular enzyme here. And this enzyme is joining these two substrates, PEP and S3P. So we kind of just use their nicknames here. And then it would produce this EPSP product. Okay, so when it is, the reason that glyphosate is very effective in blocking this pathway is because when we look at this model, so this is the enzyme here, and it's already forming its pocket because it's active, it's activated. So one of the substrates is here. So this yellow green is the one of the substrate S3P. It needs the other one, PEP, to go in here to make the EPSP. The PEP is excluded because glyphosate has replaced it. So ngayon itong green, this is glyphosate. This is supposed to be PEP. So why is it very effective? Because the binding strength of glyphosate is 80,000 times higher compared to the binding strength of PEP into this complex right here. So nandito na yung S3P, it's sitting there, naka-situate na siya, no? Naghintay na lang siya sa kanyang pair. Ang kanyang normal na pair is PEP. Pero pag nandiyan ang glyphosate, it's a very strong competitor. So nobody can beat that. And so now there is no preventing, because the fit of glyphosate is a lot better. Mas bulky kasi yung PEP na molecule. And then it happened that glyphosate is more streamlined, it's slimmer, and it fits better, so the pocket becomes tighter. And so now the plant cannot tolerate that. And so the result of that is non-selective action. A lot of plants are tolerant to it. And it's the thing with glyphosate, it is only foliar activity. There is no soil activity whatsoever. So if we're using it for weed control, it has to be paired with something that has soil activity, yung mga soil applied herbicides, right? It is slow acting, although the new formulation, the formulations we have now, the modern formulations we have now are faster acting. You can actually see activity in a few days. But it is completely systemic, and then you will see a lot of chlorosis, and then eventually it will just die. And then sometimes it can cause bleaching on some tolerant crops. This is glyphosate resistant corn. And so when it gets some stronger dose of glyphosate, sometimes it can bleach the corn, but it doesn't really kill it. So merong mga weeds na tolerant, of course, because of different mechanisms of protection. So ang mechanism of resistance to glyphosate is basically non-target site. First of all, 56 species globally have evolved resistance to glyphosate. So, but this is after many decades of use of this herbicide. It took a long time for plants to be able to overcome this herbicide. That's how difficult it is to evolve resistance to glyphosate. And then it also, the fact is, the plant cannot survive if the sequence of amino acids in that enzyme, APSPS, is changed. So right now we are seeing fewer weeds that are showing sequence mutation in APSPS. It only gives us low resistance level because the sequence of amino acids in the enzyme is highly conserved. So the only way the plant can tolerate the herbicide is to evolve resistance via other means. So exclusion of glyphosate is the big factor that has happened, that has been selected for. So reduced absorption, that has happened. Altered translocation or reduced translocation, that has happened. The recent one that is being proven is sequestration of glyphosate in the vacuole, and that is happening in a few species. And then the other big mechanism now that is conferring the highest resistance level to glyphosate in the species of weeds that evolve this resistance is increasing the copy number of APSPS, which means that the plant is now able to produce more of the target protein. So because ang label rate is fixed, we cannot apply glyphosate beyond the label rate because that's violating the label. Then it's one-to-one. So it's one-to-one interaction, right? So if you have more copy, then you have more enzyme that can do its function, even if glyphosate is applied. So it's overcoming that by the numbers. And so to just briefly look at that, there are these species of weeds that are actually sequestering glyphosate some. So they're doing sequestration of glyphosate and as a way of surviving glyphosate to some extent. So it's to a limited extent, they're doing that in nature. And then when we use glyphosate a lot, then we increase the frequency of this or the level of this sequestration. And then we actually just drive the plant to become resistant eventually. Because if they already are doing that to a low level, then if we select for that continuously by continuously using glyphosate, then we're making the population resistant gradually, just like what the video was showing. So proof that plant can actually put the glyphosate into the vacuole and become resistant is this. So in a susceptible plant, so there are two species now that are shown to be able to sequester glyphosate in the vacuole. This is horseweed, Conyza canadensis, and then ryegrass, lolium. So in a susceptible species, glyphosate actually goes, the majority of it goes to the chloroplast. So this green squares here, this is the proportion of glyphosate molecule going to the chloroplast. That's where it's supposed to go because the synthesis of amino acid is occurring in the plastid. Now in the resistant plant, in this Conyza or lolium, the glyphosate actually, so here is black one, so the black circles. This is glyphosate going to the vacuole. The amount of glyphosate growing to the chloroplast is very small amount now. And so this is now kind of how they're showing that, yes, indeed, it can happen that plants can do that. They can sequester glyphosate into the vacuole, make them resistant. Another way that plants can be resistant to glyphosate is killing the tissues quickly. So we call it kind of pruning themselves, basically. So when you apply glyphosate, then it causes rapid death of cells, and then that limits translocation of glyphosate to the other parts, because dead cells cannot translocate anything. So it localizes the damage, is what it's doing, right? And so giant ragweed, ambrosia is doing this, and actually also coniza has been shown also to do this. And other herbicides can do this too, like other contact herbicides, like PPO inhibitors, they're contact herbicides. They can also do this too. Some of those plants that are not totally killed by PPO herbicide could be doing this. They're causing faster desiccation of tissues so that there's less herbicide that can go to all the other growing points. So, when you manage that, then you just have to follow up such herbicides with something else that can completely kill what's surviving, because what's surviving is actually regrowing. Tumutubo silang pabalik. So you have to kill those ones that are growing back. So ngayon, and to complete this topic about glyphosate, yung mechanism na dadami yung target, right? Para makasurvive siya. So this is what it's showing here. It's showing Palmer amaranth, and it's showing increasing copy. So this is the number of copies here of PSPS on the y-axis. So it's going from, normally it's 1 to 2, and then now here's going to it's about 230, something like this. This is work from my lab, one of my former students doing this. Rio was still here when Shilpa was doing this. Actually, Dr. Rio Felisalas was helping in this project. And so then we see that if the copy number is higher, then the injury is lesser. But that relationship is not very clean, because there are plants that actually have low copy, like here, but then they're also injured less. So they're resistant even if they are low copy. So which means that this is, across populations and all of that. So in the field, we're actually seeing different kinds of resistance mechanisms. So how does that impact our resistance management? That means that when we manage our resistance problem, we have to be very conscious about using different tools, not only mixing herbicides, but mixing tillage and cultural practices and varieties and all of this, in order that we can take care of these others that are not in the same resistance mechanism. So there's a lot of diversity in the field. How does that happen? So there's this thing we call helitron in the plant breeders, the genomics people. They've understood that there's duplication of genes, and one of the ways that the genes can be duplicated is by these circular DNA helitrons. So the EPSPS copy increase is because of there's extra chromosomal circular DNA, and then it allows the plant to insert this EPSPS everywhere in the chromosome, and it's precise insertion. And so what we're looking at here is a representation of the alignment of nucleotide sequence, like the genome sequence of a resistant Palmer amaranth versus a susceptible Palmer amaranth. The red lines here indicate gaps in the sequence, meaning that in the resistant pigweed, there are a lot more genomic sequences here compared to the susceptible ones, and these are representing the inserts compared to the susceptible one doesn't have any inserts. So kind of just a simplified look at how plants are evolving to avoid what we do to them, to the weeds anyway. As far as target site resistance, like I said before, it's kind of rare still in resistant weeds, and the resistance level that it can confer actually is low. So hindi siya masyadong reliable just by itself to be fully resistant to glyphosate. But then there's this case where there is a double mutation in goosegrass. This is actually in Asia, I think, came from. And so this is a double mutation that is causing, so the two together is making the goosegrass highly resistant to glyphosate. So that can happen as well. Okay, now second to the last. So this is now PPO inhibitors in rice. We have some PPO inhibitors in rice, like carfentrazone. Probably in the US, we have saflufenicil as a relatively new rice herbicide. And so this is related to pigment synthesis. This is actually related to chlorophyll synthesis. So there's two pathways of pigment synthesis. So here's one that is what we call the tetrapyrrole pathway. This is where the PPO inhibitor would act. So here, this is oxyfluorphin, and this is inhibiting the formation of protoporphyrin from protoporphyrinogen. The downstream product here is, of course, you can see chlorophyll. Eventually, it's going to be chlorophyll, whether it goes through this pathway or in this MAP pathway. And so there are different inhibitors that can be used. And right now we're talking about the PPO inhibitor. So this is the enzyme that is inhibited by this herbicide, oxyfluorphin. So what happens when that is inhibited? Of course, it kills photosynthesis. But the thing is, actually, before it can kill photosynthesis, it could actually kill the plant by just desiccating the cell membranes, because this action leads to the formation of highly reactive oxygen, very strong oxidants. And so it just ruptures the cells and just desiccates the leaves even before the plant can think. So anyway, this is kind of the representation of that kind of detail of that reaction. Here's the enzyme PPO, and then here's an inhibitor that can inhibit that, and it would cause the accumulation of this. So anyway, when that happened, there are many groups of many herbicides in this for 4 families. So we're familiar with the diphenyl ethers and all of that, but here's this. And the diphenyl ethers are here. The fomesafen, oxyfluorphin, lactofen is here. And then carfentrazone is here in the phenyltriazolinone group. And then oxadiazone, which is used in rice, is here in the phenyl oxadiazole. So we have these chemistries used in rice. And rapid activity. So this requires light, because in the presence of light, that's what triggers the formation of this highly reactive oxygen atom, and then it causes peroxidation of lipids, right? It causes the leakage of this protogen into the cytoplasm, and then it converts that into protonine and makes this. So then it's contact activity, because it doesn't allow enough time to translocate. However, if you apply this herbicides in the evening and then it gets dark and there's no light, then it will actually result in some translocation, and it could actually result in better control. When the sun shines in the morning, then some of the herbicides already had translocated better, then you can have a better kill. So that's one of the kind of ways you could circumvent the rapid action. And so this is basically kind of a summary of what would happen when we apply a PPO inhibitor. So what we will see in the field is kind of rapid water-soaked appearance of leaves and kind of rapid desiccation, kind of like what this is here. So in this case, it's like totally killed this broadleaf species here. This is rice. We're looking at carfentrazone, and some of the rice may be affected a little bit, but it's really tolerant. So carfentrazone is labeled on rice. This is grain sorghum, so it's really wadded and twisted by carfentrazone. This is corn. It has some tolerance, of course, but then where you sprayed it, you can see. So you can actually tell when, like how many days before. If you're familiar with corn development, then you can actually tell when the farmer sprayed the herbicide, because it will show like how much growth had happened. So you can see how contact it is. That, and then this is showing some tolerant species. So peanut is sickle pod is tolerant, peanut's tolerant. This is Johnson grass. It's tolerant, but it's still kind of damaged. And then this is saflufenicil. It's kind of a stronger version of a PPO herbicide. And this is also labeled in rice. And it has stronger grass activity, as you can see, and also stronger activity on broadleaf species. But rice farmers still has to be careful, because the margin of tolerance is, you cannot kind of abuse the margin of tolerance because it's not going to be as forgiving as the other PPO herbicides. So this actually broad spectrum. This is a broad spectrum herbicide. It has soil and foliar activity. So it is applied, it can be applied pre-plant for some crops, pre-emergence for some crops, and then foliar as well. Now, resistance to PPO. So actually, Dr. Salas Perez Rio, her dissertation is about this, resistance to PPO in Palmer amaranth. So a lot of the database that we had generated actually came out of Rio's work in my lab. So what we know now is that there are 10 dicots and 4 monocots that have evolved resistance to PPO herbicides. And primarily these are in the US, and I only saw like 2 cases in China. And then a lot of pigweed species and these. So what we've learned so far is that there are these mutations, primarily the deletion of a codon in 210 position, glycine codon. And that is the most common mutation. And then there are these other mutations that are also occurring. And so that is what's making in terms of the amaranthus resistant. And then what we also learned is that these mutations can accumulate. So what we've seen so far is that 2 of these mutations can occur in the same plant. So then you can imagine now there are 2 mutations in the same plant. But what we've learned though is that if there are 2 mutations in the same plant, each mutation will be sitting in a different allele because Palmer amaranth is a dicot. It's got 2 alleles. And so then if the mutation occurs in the same allele, the plant cannot survive unless it is a heterozygous, meaning it has to have another allele that is not a double mutant, because the double mutant is not functional. So it's physiologically dead. And so we need to have an active allele in order for the plant to survive. So there's kind of fitness penalty going on around here. But the lesson here is that there's also a great diversity, even in the PPO mutation profiles, and it can alter the tolerance level, the resistance level. And it can also alter the way that we would mix herbicides and the types of herbicides that we'll be mixing. Okay, the last group would be this one. This is going to be group 15, which is still fatty acids, but now it's long chain fatty acids. And then the herbicides in this group are actually soil active herbicides. So they're soil applied. So remember we saw this earlier, the ACCase enzyme is inhibiting this point here that is making this 18 carbon chain. After these are made, some of these actually are transported to the cytoplasm, and then they are made into longer chains like 30, 20 something. And then these are the fatty acids that are going to make waxes and suberins and cutins, so structural components of the plant. And so of course, if you remove that, then you kill the plant, right? So what is the effect of this? So what are the herbicides that are there are these herbicide families that are inhibiting that particular enzyme, that elongase, and so chloroacetamides are here. And so in the rice market we have butachlor, pretillachlor, chloroacetamides that are cousins of S-metolachlor that are used in rice. And then this group of herbicides used in many crops, like for example, S-metolachlor alone is used in more than 50 crops, used in all sorts of crops all over the world. So this is from plantation crops to turf grasses to fruits and vegetables. Very important group of herbicides. This is one of the example of groups that we actually need to conserve as much as we can, because this herbicides allows us to manage most of the other herbicide resistant weed problems that we already have. So we can use this in the overlaying of residual herbicides to overlap them so that we can achieve more or less season-long control and minimize hand weeding later. And so these are very useful in that regard. And also it is difficult for plants to evolve resistance to this herbicide group, because where it acts is is a difficult place to evolve. So it's kind of conserved also. And so these are primarily, well, we could say that a lot of these are good on annual grasses, and then also small seeded broadleaves. And what we will see in the field is either lack of emergence, so they're just gone, or those that are tolerant, a little bit tolerant and can emerge, they are stunted or wadded up. So anyway, you can see that. So see this field here used to be full of grasses, but then it's totally clean. And then soybean is tolerant, okra is tolerant, corn. So there are many crops that are, depending on the herbicide, okay, so tolerance would differ slightly depending on the herbicide. Peanut is tolerant to this herbicide. Then this is grain sorghum that has received higher than normal rate, and then now it's wadded up and stunted. So grasses that are sensitive or low tolerance will be looking like that. So then resistance to this group. So far, there are 2 dicots and 11 monocots, meaning 11 grasses that have evolved resistance. And the dicots here would be, one of these would be the Amaranthus group. Well, tuberculatus and palmeri, and then many grasses, 11 grasses. And there's understandable, because the major target of this herbicide are grasses. They're the ones that are generally susceptible. And so then because the target site is more conserved and is complex, the resistance mechanism of the herbicides that are becoming resistant is non-target, meaning it's outside, something else that they're doing. And what this something else is, we found that in Palmer amaranth, in my lab, we did a lot of work on this one, and we found that the resistant population has increased the expression of glutathione S transferase. So this type of enzyme is the enzyme that acts downstream after the cytochrome P450. So remember, we mentioned cytochrome P450 earlier. And so after the P450s have broken the herbicide in phase 1 detoxification, then this type of herbicides, conjugators, would act on the metabolites. Then the metabolites would get tagged or attached to this to glutathione via glutathione S transferase, and then tagged for moving into the vacuole for further degradation or just for deactivation, right? And so when the activity of this enzyme is increased, or expression of this enzyme is increased, that means it's increased conjugation into less non-toxic metabolites. And so that's in rice. There is actually barnyard grass, Echinochlora corsgali, that has evolved resistance to this group that is used in rice. And so that is kind of the group of herbicides that we could cover with the time we have. And then really going back to resistance management. And so I have just listed here the major points that we would want to consider in our resistance management efforts. And this really is also a recap of what the video was talking about earlier. And so I believe this is a good stopping point, and then we can proceed with discussing any questions that you would like to discuss.
Thank you, Dr. Nilda, for that very insightful presentation. Thank you, Doc.
Hi, Janina.
Hi, Doc. This is Janina Signawan, and with Edward here. Thank you, Dr. Nilda, for that insightful presentation. Actually, hindi lang siguro sa farmers or sa students or sa mga local government units, even us in the industry, very informative po yung presentation. And actually, medyo may mga new words, new findings din po kami na nalaman today. Actually, Doc Nilda, we are today with our previous speakers nung last 4 sessions natin, with Dr. Magsino, Dr. Dindo Donaire, Dr. Nina Limaana and Dr. Rio Salas Perez. Actually, nandito sila. Some of the questions in our Q&A tab were actually answered by them, and it's a medyo madami. Regarding ating Q&A tab and yung ating chat box and even yung ating Facebook page, marami pong questions na itanong. At actually, may mga sagot na rin po sila. But
That's good.
yes, thank you, Doc Nina and Doc Rio. Actually, nakasagot na rin sila. And si Sir Hill din po, nandito rin siya. So, dito sa ating Q&A portion, let's try to be interactive. Yung mga questions natin, hindi lang si Doc Nilda yung pwedeng sumagot. Pwede rin yung ating mga previous speakers from our previous sessions and webinars sa Crop Life. I'd like to point in our first question here.
Sige. Ay, pwede ako sumingit, Janine.
Yes, sir.
Merong napakaraming question. Mukhang very interested talaga sila sa presentation ni Doc Nilda. Ang question nakita ko, Miss Jen, na medyo paulit-ulit. Pwede daw bang humingi ng presentation?
Sure.
Opo.
Actually, I was thinking about that. I could send this in a PDF form. And then also I was thinking na, kasi itong mga bago naming mga publications coming out, maraming mga open access. I could actually also send everything that we have relative new. Padala ko kay Janina at kay and then Janina can distribute to whoever is interested in getting copies.
Yun. Masasagot na mga assignment.
Marami na pong nasagot, Sir Edward, na mga 20 questions na ba yun?
Hindi lang.
PDF?
Oo, pwedeng PDF. Thank you, Ma'am Nilda.
Thank you, Doc Nilda. Ayan, Sir Gil, Ma'am Rio, and Ma'am Nina. Okay.
Oh, I'm seeing Rio.
Yes.
Kumusta, Rio?
Parang ito po kasing session 5 Dr. Nilda is a culmination activity of this HROC webinar session ni Crop Life HROC Philippines. And we're very thankful na kayo yung aming last and mga ating previous speakers ay nandito rin po present. So Doc Nilda, I'd like to first ask you a question from Sarah May Perales. Although sagot natin ni Doc Rio, baka doon sa mga ibang hindi nakakabasa ng Q&A tab kasi yung iba ay nakakabasa lang ata doon sa chat box. From Sarah May Perales po, is fluazepop butyl and glufosinate ammonium compatible when mixed together?
Yeah, they can be kasi they're not listed among the antagonistic ones. Yeah, they can.
Okay, thanks.
Yeah.
Thank you, Dr. Nilda. And okay, may foreign participants daw pa tayo. So okay, I need to be in English informed tayo dito. But for the sake of all the other participants, not just Filipino participants and foreign participants. So from Sir Froilan Cruz, actually this is the question from the previous webinar din na narinig po namin. How long does a herbicide stay on the land? Is it safe a week after spraying any type of herbicide to drop off the seeds for planting? Actually, Doc Nilda, from the previous session, we also received this kind of question. Very common po.
Oh, so the question is how long
Does
you can
herbicide stay on the land?
Right. So the best basis for that actually is to look at the label, because on the label they actually have specified rotation intervals. So it because then this one depends on a lot on what the herbicide is, because well there are herbicides, for example the ones that we talked about today, like glyphosate, that one doesn't have soil activity at all. So you can apply glyphosate like now and then you can wait a little bit for the spray to dry up and you can plant your crop tomorrow, depending on what the size of the weeds are. So and then for example the selective grass herbicides, a lot of those do not have soil activity at all, so you can plant your crop like immediately after. But then one of these, maybe clethodim or cetoxidem, actually requires like 1 week interval before planting something like grain sorghum, because it has some residual activity. It varies a lot by herbicide. Now, strong residual, let's say, for example, atrazine. So atrazine is a corn herbicide. That one has a very strong residual activity. So that the only thing that you can plant after you apply atrazine is corn or grain sorghum. And other crops, you'll have to wait the next year to be able to plant them. So that is some examples of how, you know, in that question, you'll have to look at the label depending on what herbicide that is.
Thanks, Doc Nida. And for the other panelists po, they can bat in anytime. So basically, it depends on the herbicide. So yun talaga yung sagot natin. It really
Depends on the herbicide and the crop, because like I said, the herbicide atrazine, for example, the crop corn and sorghum is resistant. They can be planted like immediately, but then no other crops can because they are susceptible. So yeah, so both depending on the tolerance of the crop and then the strength of the residual activity of the herbicide.
Yes. Thanks, Doc Nilda. And another question po. Actually, nasagot na to kanina ni Sir Edward, but there's question from Sir Reynaldo Tabulo is, in the challenges of translocation, would you recommend to use adjuvant sticker in spraying herbicide? Actually, this is on the first 2 slides niyo po kanina, Doc.
Right. Yeah, adjuvants can help with retention, spreading of the spray on the leaf, distribution of the spray on the leaf, retention of the spray, and then entry of the herbicide across the cuticle. But then translocation, it's a different story now because that would depend more on the herbicide chemistry.
Okay.
Yeah, because then it depends on the lipophilicity of the herbicide and all of that. So yeah.
Yes, sir. So yes, Doc. So basically, you can add adjuvant or sticker. It depends also on the herbicide, on the target weeds and crop, right?
Oh, actually, the rule of thumb is if you're applying a foliar herbicide, you have to add an adjuvant. There are a few exceptions to this rule. For example, let's say glufosinate, it has a built-in kind of, but we don't add because the manufacturer had already built it in, and that's the reason we don't add. But actually, the practice here is for tough weeds and tough conditions, bigger weeds, it actually is beneficial to add an adjuvant even to glufosinate. So the popular partner adjuvant for glufosinate here is a sprayable ammonium sulfate. So ammonium sulfate is actually fertilizer, but it is also formulated as a sprayable version that can, you know, be mixed with herbicide, and it improves the activity of glufosinate a lot. It even could improve the activity of glyphosate also a lot, depending on where you are. So yeah, so the general rule is use adjuvant for foliar application, right?
Thanks, Doc Nilda. And of course, niyan tayo sa Zoom, merong mga participants. Sir Edward, I think we have questions also from our Facebook page.
Naipangutana from Zamboanga. Then, of course, thank you, Sir Mark from FPA po ang question. So thank you, Sir Mark, for joining. Question is, Doc, why daw po rice plant is not affected by ALS inhibitor herbicide?
Right. Well, no. Some ALS inhibitor herbicide can kill rice, so depending on what ALS inhibitor that is. Right. Like, for example, that imazethapyr, it can damage rice there. So okay, the ALS inhibitors that do not kill rice is because rice is able to metabolize the herbicide. So in many cases, and Nina will probably agree to this and contribute, in many cases, the way that plants can survive a herbicide is by detoxifying it. And so differential detoxification. So for example, is not necessarily even ALS. For example, ACCAs. So ACCAs inhibitor is grass herbicide, right? So let's say, what is an ACCAs herbicide in let's say cyhalofop. Okay, so cyhalofop is an ACCA's herbicide. It's recommended in rice for grass control. Crying out loud, right? Grass control in rice. How does that happen? Controlling grass in a grass crop. So how is that possible? Well, the susceptible grasses, barnyard grass, whatever, sprangle tops, those grasses cannot metabolize cyhalofop fast enough. So they're killed. But then rice can actually, what is that? So one of the reasons is there's less entry, less penetration of cyhalofop into the rice cuticle. And the other one is that it's faster deesterification because, well, butyl, it's cyhalofop butyl. But anyway, faster cleavage of that butyl chain, and then oh no, not faster, slower, because it becomes active in the plant when you cleave the ester chain, and then it becomes its acid form, and then it gets translocated, then it becomes herbicidal that way. So if the cleavage of this side chain is slower, then it remains like less active for quite some time, inactive for quite some time, and then it gives the plant a chance to metabolize it if it can, something like this. So yeah, differential entry of the plant and differential detoxification into the plant and differential detoxification of the herbicide is why, this could happen.
Thanks, Doc Nilda. Very, yung pagkaka-explain niyo pa talagang malinaw kasi talagang yan yung very most commonly asked questions why ALS herbicide. Because it's usually ALS herbicides in the Philippines are usually used for rice and it does not cause any harm to rice plants. But as for this ACCAs nga, grass versus it can control grass, but it's also safe to grass as rice.
Yeah, grass and grass.
That's
Right,
always
right.
the common question.
There's another ALS analog, kind of parallel to that. So we have this sulfonylurea we call nicosulfuron. It's a sulfonylurea, so it's an ALS inhibitor. It is labeled for corn, for control of grasses in corn. So see, so that's kind of the same principle that you get too. So corn can metabolize it, and then the susceptible grasses cannot.
Yan, kung ano gagawa ng technology.
Right.
Follow up, Doc, not related to that, about does soil types affect the efficacy of herbicides?
Oh, that is a major
Doc.
Right, that is a major factor in herbicide activity is soil type. So anything that herbicides that have soil activity, their label rate is in a range, and the range is because it is adjusted according to the soil type. So it's a coarse kind of classification. So we have classification of heavy soil or medium soil or light soil, and this really pertains to clay content. So light soil is more silt, more sand, less clay, and heavy soil, a lot of clay and sticky, stuff like that. So more binding sites for herbicides. So in many cases, the herbicides that are soil active are labeled at a lower rate when we are in a sandy, silty soil, and at a higher rate when we are in a clayey soil. Now, if we violate these rates, we can kill our crop because some crops do not have a really big margin of tolerance. So if we overdose them in a light soil and use the wrong rate, then the crop is damaged. So that's why also another principle in the resistance management video, right? Use the right rate and then apply that to the correct stage of the crop and the weed. So
yes, always follow the label recommendation. Yan po talagang ating laging paalala. Last na po, Doc, for the technical questions, and then we'll follow up with some suggestions and some political questions. Last po is, pwede po bang i-mix ang glufosinate and glyphosate chemical to control annual and perennial weeds? Will there be any resistance that will happen?
Oh, actually, so that question is actually kind of it comes at the right time when we are actually doing this, because so now we have these crops with stacked traits, right? So we have crops that have resistance to glyphosate, glufosinate, HPPD, and MPPO. So sometimes our crops have like 4 different herbicide traits put in them, and so that's common now. And a lot of the soybean and cotton in the US, or corn, actually carry resistance to both glyphosate and glufosinate. So because of that, then the growers can apply a mixture of glyphosate and glufosinate. Now, actually, this is kind of something that farmers could also learn by kind of trial and error, because this mixture can be optimized. I believe that you could achieve the best of both their potentials, and so in the right mixture. But we don't really have a really good handle on if there is an optimum combination of these, except for following the label rate. That, okay, if we label here, we're dealing with let's say if we label 0.75 kilograms acid equivalent of glyphosate, and then 0.4 kilograms active ingredient of glufosinate, something like that, and just following the label of each, it still works, and it actually would help. The presence of glyphosate there could help improve the control of perennial weeds, because glyphosate would translocate a little bit more. You just don't want to overdo the glufosinate, because we know glufosinate is more faster acting, so it actually has limited translocation. And then also, if you apply that on the optimum time of day, the best light situation, and all of that, it becomes more contact than systemic, because of course, again, the production of the strong oxidants, right? Because it's light dependent, then it becomes more active, then it could limit. So then what it can do is it can limit the translocation potential of glyphosate. So that's why I was saying that right now the farmers are really just following the label rates, but we don't really know if we have a mixture of perennial and annuals, if that is the optimum combination. So you guys can do research on that and
Yes,
determine optimize this combination, because now we know that we can combine them, but we just see if the combination can be improved.
Okay, but this just to remind that glufosinate and glyphosate are both non-selective
Yes,
herbicides.
they are both non-selective. That's correct. But the combination of the two is because glufosinate can actually take care of glyphosate's weakness, because glyphosate has weakness in some species. So, for example, there are what we would call, although it is non-selective, there are species that have some tolerance to glyphosate. They're difficult to kill. And so example of this would be the morning glories, the weedy relative of sweet potato. Those are difficult to kill. They cannot be killed by glyphosate as soon as they get to a certain size. So if you have glufosinate in there, then you can have a good control. So good control overall.
Yes, thanks, Doc. I think it was also mentioned by Doc Hill in his presentation last webinar for the corn weed resistance management about this different tolerance of different weeds to different kinds of non-selective herbicides. Follow-up, not really follow-up question, but not really a controversial question, but curious question from our participant, Mr. Ronald Peralta. Hello po, ma'am, sir. Based on my observation, one already developed a GP corn glyphosate tolerant and it was successful. My question is there any possibility of developing the same technology to rice? Because in the Philippines, rice is our staple food. Can any company develop rice glyphosate tolerant or rice herbicide resistant species?
Glyphosate tolerant rice. Okay, so let me share a story. So the only way that we can have glyphosate tolerant rice, and the reason that we are able to have herbicide resistant rice, is because that technology does not involve putting a foreign gene into rice, meaning it is a non-transgenic technology. So the public, the consumers, they do not accept genetically or transgenic rice. They do not accept transgenic rice. And this reason is what killed glufosinate resistant rice. Actually, in my first 2 years of being faculty, one of my research was about the first batch of glufosinate resistant rice. We were in the, testing phase in the field, and demonstration, and, field plots and all of that. We were all so excited. And so this was a story, long ago. But anyway, it's kind of a lesson about how difficult it is to put herbicide resistance straight into the rice market, because it's not, there are many potential technologies of herbicide resistance we can put into rice, but if it is transgenic, then you can forget it, because at that time the European market is not accepting GMO rice. And so then in all of their very detailed testing, so then they detected a contaminant, very, very, very low. I don't know, a percentage that's almost like next to infinity, but it's detectable. So when you detect something, then it's a detection, right? And so it's contamination, and so then it's a basis for arguing that, oh, this is contaminated, and so we're going to reject it. So then it made the industry realize how difficult it is to segregate normal rice from GMO rice. And that became like an impossibility, and so then they just have to scrap the technology. Now we have HR rice, but it's not transgenic.
It's not transgenic, yes, not like a GM crop. Yes,
right, it's not a GM crop, right?
Thanks, Doc Nilda. And for the rest of our panelists, actually, this is just a suggestion from our participant, from Geman Asohedo. Siguro, ma'am, sir, our government should venture private sector or technical person to conduct a seminar for our beloved farmers to tackle these topics, to educate the low level of education. May mga farmers lang po kasi na gumagamit lang sila ng herbicide na gamit ng katabi nila or kapitbahay nila. Yan. So, actually, challenge po ata ito sa atin, especially to our CropLife and academe, our local government units, to the companies, industry, na pagtulong-tulungan na makakonduct ng ganitong kind of training for our beloved farmers.
And actually that has got to be integral na. Actually, it has to be integral in how we do agriculture. Is we have to have trainings targeted specifically for farmers, and then we have to have trainings targeted specifically for, all our crop advisors and companies are training their own people, and the fertilizer companies are also training their own people. But then we also have people from the Department of Agriculture, crop advisors, our extension workers. They also need to be trained at certain levels, right? And so then they can help eventually to train. So it's kind of like training the trainers. We've always had this, even when I was young. We've already been doing this, training the trainers. What is that? Farmer to farmer training. This was the key word when I was young, when I was in my teenage years. And it still should be, it should still be a keyword that we have to carry today. Farmer to farmer training and expert to expert training, just passing on information down the line and spreading it out and lessening, reducing the load for everyone. In training, we do need that. So this is actually not anymore a luxury. This is actually a necessity for us to be able to continue to do agriculture, to be able to survive, because our farming now is complex. It's complicated now. And especially if you put in climate changes, etc., that that throws in a big wrench. And so we have to actually consider this is not anymore just something that we do because we want to feel good. It's got to be
Yes.
something that we have to do
Yes.
because we need to survive.
Yes, yes.
Yes.
We agree to that. Yes, Sir Hill, you have something to
Doc
share
Hill,
po?
yes po.
Yes. Kay Doc Nilda, no? Alam mo, Doc Nilda, in the Philippines, marami tayong kinakandak extension and training sa mga farmers. But then, you know, I know you know this already.
Yeah.
One of the main challenge sa farmers training ay yung kanilang attitude or behavior.
Mhm.
You know, so difficult to to educate them. They will say yes in front of you, but at the back, they will still use what they want to use. For example, most of our farmers are brand loyalists. What are we going to do with those farmers with brand loyalists?
Right.
So if they know that this product in rice, for example, is very effective, so
That's
I
all
will
they
use it.
use. Yes.
Who
That's true.
cares? I will use this one. Why you know what I mean? It's so difficult to to emphasize to them the value of MOA rotation. So I think we need a social scientist said we need to protect the product which is already in the market. They're really very, very useful, but they fully understand the integration of the different approaches. But we know that herbicide is one frontliner to defend against weeds. That's the problem. Yeah.
That is true. Yeah. It's you should have a social scientist and very patient and personable people to to be always there with it's gonna be village by village kind of thing, and establish relationships, establish trust. Gotta it's really is it really is banking on relationship building, so the farmers would kind of feel um connection. Because a lot of times the farmers would, yeah, like you said, yeah, they would listen to you, and then and then after that they'd go their own way and do the same thing. So but if they feel more connected and there's a closer bond, then they would feel a little bit more sympathetic. Maybe they would feel
Yeah.
sorry for you because you always go there and train them, and then they're not, their conscience will bother them, and then they will follow. But anyway, but we do what we can, right? So
Yeah.
we just do everything we can, and that's all we can do. Like you said, there are many things that are out of our control, and the decisions of people we cannot make for them.
One more question, Doc Nilda. Isa lang. One more question. What are the challenges ahead of us, or what are the reasons why there is still unclassified group, HROC group? Is
Ah,
is this is unclear phytotoxicity processes is one of them?
no, the persistence doesn't have anything to do with that. It's all about understanding what it does in the plant, because the way we classify them is we classify them according to where they go or what they do. But then some of these herbicides, you apply them, and then it's hard to pinpoint what they really do exactly. Or do they really exactly do. And so meaning that probably the manufacturer or the whoever discovered it didn't get the chance or the opportunity to study in detail where it goes, because this entails following the molecule. It entails electron microscopy, tracers, all of these kind of detailed studies that are good for PhD students. So, no, but actually, yeah, it takes a lot of effort and time and just a lot of detail to understand the mode of action of some of these herbicides, and
Thank
that's
you,
why they remain.
Janilda. Yeah.
Yeah.
Yeah, I think Dr. Nia Di Maano here and Dr. Perez are one of the drivers also during their PhD, ba? Yung mga
Right.
na-discover nila based from their thesis. Mukhang malaki ang
Yeah.
na-contribute po based din sa presentation ni Dr. Nilda.
Yeah, a lot of our database, Rio had participated in building them. So, for example, the copy number, APSPS. So in Rio's masters, we discovered for the first time the increase in APSPS copy in a grass. And so that was in Lolium. That was from Rio's work. And then in her PhD, with her work, we discovered the PPO resistance situation for the first time in Palmer amaranth. So anyway,
Yan.
so yeah,
We have a good lineup po talaga ng presenters, Doc Nilda. Bago yung culmination activity ng session 5, we gathered
Yeah.
din para naman talagang sure na sure po tayo.
Yeah.
Actually, we still have lots of questions po in our Q&A tab, but due to time constraints, we will try to collate that and send that to you if we can also answer, and to our panelists if they can answer via email. At this point in time, Dr. Gila, we will ask if you have questions prepared for our participants or you don't have po.
I don't know. Would they remember?
Yes, because they should remember, Doc, because we have a cash prize for that. We have like a 500 peso GCash for those who will be answering from Facebook and from our Zoom. So Doc, if you can share po, and then we will by the way, just reminder for those that who will win, please subscribe. Ito yung ating criteria. Subscribe to Crop Life Philippines YouTube channel, like and follow our Facebook page. Hindi lang namin gusto ng maraming subscribers, but we also would like it to like and follow para updated po kayo sa ating activities, sa ating mga webinars. And during the Q&A, the first person to answer each question wins, and there will be 3 winners from Zoom and 3 winners from Facebook. Well, winners will be announced and notified during the webinar and FB Live, and Crop Life representative will be in touch with you. Prizes will be sent through GCash po.
Okay.
Yes, Doc Nila, if may questions po kayo para may chance din po manalo yung ating mga participants po today.
So okay. Do I just say it? Like
yes.
Now?
And then we will monitor po dun sa ating chat box and Facebook page.
They will probably not remember this, but this is a new term. It's a new term that I introduced that is related to that is actually the mechanism that allows Palmer amaranth to increase its EPSPS copy number. So what is the name, the technical name of the circular DNA that is allowing increase in APSPS copy, inserting these copies to all the chromosomes?
Oh my God. May makakasagot kaya. Oh, wait, may sumasagot po.
Masagot talaga. Oh, somebody actually answered that.
Doc Nilda, ibig sabihin, nakinig po talaga sila ng mabuti.
Pero yung Janina, yung sa Facebook, wala pa ha.
Yung sa Facebook, mukhang naliligaw pa, Sir Edward. Mukhang inaano pa. Sinesearch pa sa Google.
Oh my goodness.
Doc Nilda, tama po ba na ang answer ay Helicon?
Yeah.
That's okay. I think we have 3 winners. So,
you have a winner.
Yeah, so contact po kayo ni Crop Life, and then we will one person from our team is monitoring sino po yung mga first 3 winners po sa ating Zoom and Facebook page. So, ang sagot po ay Helitron.
Mhm.
Meron na rin sa Facebook. Ano?
Yun, talaga namang hindi nagpahuling
Meron na
atin.
rin.
Usually po kasi
Very
Doc
good.
Hilda, yung mga nasa Facebook page, yun yung ating mga farmers, distributors,
Ah,
yung connect sa Zoom.
okay.
Mas convenient sa kanila sa Facebook.
Ah, okay.
Okay, so congratulations po sa ating ma-announce na winners. Another question po
Good.
siguro, Doc. Para naman mas maraming winners, mas masaya.
Para mas maraming winners. Okay. So, this one is easier. So, how do you judge if many of them would get the correct answer?
First three to type
It's by how fast they answer.
Yes.
Okay, that is cool. So, all right. So this one is easier. So what are the 3 amino acids that are inhibited in production by the ALS inhibitors?
So dapat tatlo, bawala isa lang.
Aha, dapat tatlo.
Pag one answer, hindi po valid.
Right.
Mukha ang bibilis.
Marami
talaga.
sumasagot.
Ang daming sagot. So they know this topic. Very good.
Ayan. So yan, namin yung sumasagot.
May pabilisan lang pala ito.
Pabilisan. At saka po, hindi pwedeng i-search sa Google agad-agad kasi kailangan natandaan. Kailangan
Right,
Ano, o nga pala, reminder sa mga sumali, nag-type ng answers, may it be sa Zoom, may it be sa Facebook. Baka minsan nakita nyo dun sa live chat na kayo yung nauna. Ang susundin po natin is yung admin. Para fair.
Okay.
Diba, Jen, no?
Alright.
Para it will balance out yung speed ng internet. So,
Yes.
antayin po natin yung
Okay.
announcement from CropLife kung sino po yung official winners.
So, Doc Milda,
Okay.
yung mga sagot dito is isoleucine, leucine, and valine. Tama po ba?
Yes.
Okay.
Mhm.
Actually, may mga answers din na iba, pero yun po yung tamang sagot, Doc, no?
Yung
yun.
Yung tatlo. Yeah.
Tatlo. Okay,
Isoleucine, leucine, and valine, right?
Yan, mukhang maraming masaya. Okay, webinar. Doc, last question po para naman 3 questions tayo. Meron tayong 9 winners para actually tama ba sir? 9 winners, 2, 4, 6 yan. 12 pala.
Okay.
Last
So,
question.
last question. Okay. So, we have, so like in the last topic, we have some chloroazetamides, group 15 herbicides that are used in rice. And then we have 11 grass species, evolved resistance to that, which is expected. But then we have only 2 so far dicots that have evolved resistance to chloroacetamide, in which case this would be metolachlor. And so what is one of this, what species, name one of the dicot species? There are 2 species that evolve resistance, right? So just name one of these species. And then what is so it's a pair. So what is the species? And then what is the resistance mechanism? What made it resistant to chloroacetamide? Because this could happen in the rice too. I mean, in the rice situation too, right? If the farmers use chloroacetamide continually in rice, not rotating, it could also happen there. So knowing it ahead of time could be useful.
Yung question, sagot po, Doc Nilda, very diverse, magkakaiba.
Right.
Ano kaya ang
Magkakaiba.
tamang answer? Actually, may mga
Okay.
sumasagot ng Amaranthus palmeri, Amaranthus tuberculosus,
Yeah, both are correct. Amaranthus palmeri, and so do you have like half price? Because if they only get the species, then they only have half the price.
Yeah. So, ano pong tamang sagot?
Or we could the species is either Amaranthus tuberculatus or Amaranthus palmeri or Amaranthus. Yeah, the mechanism is increased expression of GST, which is the conjugating one of the conjugating enzymes, GIS. GST.
Hmm.
Yeah. Well, we can
Sagot.
ask another.
Yes.
We can scratch that and ask another.
As another po siguro, Doc, kasi mukhang walang dapat dalawa yung sagot niyo, Doc.
Wala nagagawa sa correct pair. Right.
Walang
All right.
ano correct pair, Doc.
Right. Okay. So let's see. So this one. Why is rice not killed by cyhalophobe, but barnyard grass is?
Talagang pang matalino po yung mga questions natin for today.
Kasi tinanong mo yan kanina. Sino
Yes,
nagtanong
opo,
yan
talaga
kanina?
dapat natandaan nila.
Right.
Ayan po, Doc, ang dami. May sumasagot ng detoxification, faster metabolism of cyhalophobe in rice versus barnyard, detox mechanism, detoxify. Tama, Doc?
Yeah. Detoxification
talagang
or metabolize.
Naalala po talaga nila, Doc.
Right.
Ang sagot nyo kanina. Okay, so congratulations to our winners po, and we will notify, CropLife representative will notify you if you are the lucky winner for our 3 trivia questions po for today. Thank you, Doc Nilda, for the presentation, and then sa ating mga panelists also for answering questions during your presentation and during this Q&A portion po. Sir Edward.
Yan, syempre.
Thank you very much.
Ay, thank you. Thank you, Doc Nilda. And of course, sa mga panelists, maraming salamat for helping us answer lahat ng questions, may it be technical, practical. Thank you sa mga panelists and of course kay Dr. Nilda for answering. Yan, thank you. And isa pa is thank you sa 200 plus.
I know.
Okay, 200 plus na participants natin. And I think this would be more than, madami din, Doc Nilda, sa Facebook Live. Na ito ang point, hindi sila bumibitaw, Doc Nilda. Talaga mula umpisa hanggang natapos, they're all here. They were.
I know, and I just apologize. It was kind of fast because we have this, topic to cover and only a certain amount of time. So, but hopefully you remember some of it, and it's great to see a lot of you guys. And then Nina, it's great to see you, Rio, and Dr. Hill, good to see you.
Thank you, Doc.
And thank you.
Good evening po ulit. Thank you talaga, Doc, for really may request daw, lecture again. So moving on sa ating next part ng program.
Okay.
Of course, I would like to call on yung aming, sabi nga ni Doc Nilda, isang ating commander-in-chief. I would like to call, of course, ang ever supportive, ever active po na Executive Director of CropLife Philippines, si Sir Ramon or Mon Abadilla. Sir Mon, magandang umaga po.
Hi, magandang umaga sa inyong lahat. Thank you to Dr. Nilda for a very informative session. No? I think it's already 11 o'clock in the US, in Arkansas.
Yeah,
We
11.
Yeah, I was looking at the time here and the time there is almost 11. So we'd like to thank you very much for gracing our last session. Not the last, but the last session for this year. And we are trying to come up with a lot more ideas on how we could continuously propagate education to our farmers, to our stakeholders, because that's very important for us. No? That's the only way we could actually influence our capability to produce more food and have sustainable farming. Okay,
That's
so
true.
I guess I'd like to, since we have the other panel, other lecturers that we had previously, Dr. Rio, Dr. Nina, and Dr. Hill. Since this will be our 2022 culminating session, we'll try to have an event so that we could maybe have a face-to-face, assuming we are allowed to do it, and be able to honor you all, no? hope, but not. I don't think we can go to Arkansas and bring it there. But maybe we
Yeah,
can
no, right? Yeah.
we can
Well,
just give you our special recognition. I'd like to thank the HRAC team. This team has actually been very aggressive, not aggressive, but more proactive in
proactive,
terms of
right,
delivering these sessions.
right.
There are 5 sessions that they targeted, and I was surprised that we were able to finish all of them. My hats all to them, and I'm very proud that you guys have been able to deliver these 5 sessions, and I know it takes a lot of effort with your work in your companies and trying to do this sideline for CropLife Philippines, no? So thank you, and I guess the main what do you call this? The main receiver of the service is really the farmers and the academe and the people that has been attending. The attendance did not drop off
I
up to the
know.
end. No, I
That's
think they're dropping
amazing.
off now because I'm the one speaking already.
I know.
They're not gonna get anything from my side, but the lecture was really very informative and a lot of information. The participants stayed on. Usually, when we get to this point, we had a big drop off, but now we did not know. We were Bo, uh, our communication expert, has been monitoring, and he was happy that no one is dropping off. Uh, Again, thank you everyone, and I guess in our own little way, we were able to deliver these modules, but we are working hard on trying to get more ideas on how we can deliver our message, deliver our promise to the farmers, to the stakeholders, to the academe, so that they can have access to the technology that our companies, our members, are able to develop more towards sustainable farming. Thank you very much, everyone. And again, thank you, Doctora, um,
You're welcome.
for your staying on late up to this moment. Thank you. Thank you very
It's a
much.
pleasure to see you, Dr. Ramon.
Okay, thank you. Doctor, I'm not a doctor. I'm an engineer.
Oh, well.
General.
You're
Ayun.
Thank you very much.
Ayun, thank you. Thank you, Sir Mon. So
Yep.
again, talagang even si Doc Nilda is coming. Sabi nga ni Sir Mon, anong oras na, of course, in the service of the Filipino people. Maraming
Right,
salamat po.
right.
And maganda yung message ni Sir Mon. Magtatanghalian na, just
Yes.
consider yung kakainin
Okay.
natin
Okay.
is of
you
course
Yep.
coming from our farmers. So talagang let's make sure na we
Right.
deliver this message to them. And then post natin ngayon yung mga winners, of course, ng ating games na makakatanggap ng GCash. Iko-contact
Okay.
natin kayo. So pasuyo po, Sir Bo.
All
Kung
right.
pwede po.
I'm gonna leave you all.
Ay,
So
thank you po.
Dr. Limon,
Thank you.
Rafael, Edward, good job. Okay,
Thank you.
you all
Thank
guys
you.
Thank you.
did a
Thank you,
great
An
job.
honor.
All right. You have
Next
a great
time
day.
po ulit.
Okay, bye.
Have a nice rest.
All right. Bye, Janina.
Bye
Bye.
po.
Bye.
Good night
Okay.
po.
Bye, Rio. Okay.
Thank you po. Thank you. From our admin, i-message na lang po yung mga winners. So,
Mhm.
madami tayong winners. Actually, I think more than 12 pa, Ma'am Janina. So, daming winners. Message po kayo ng secretariat natin for you to receive your GCash. And again, thank you everyone for participating sa 5th session ng ating Alam Mo Ba webinar series on the herbicide mode of action and herbicide resistance management. Yung e-certificates nyo po will be emailed to your registered email addresses. And then may survey po. Ayan, parang ganyan siya. Lalagay yung pangalan nyo, sign ni Sir Mon. And then of course, kindly answer the survey po that will pop out after the webinar. It will really, really help us in terms of improving do sa ating mga next na mga activities. Don't forget, sabi ko nga, sabi din ni Ma'am Janina kanina, don't forget yung ating social media accounts, Facebook, YouTube,
and ating podcast. Huwag niyo pong kakalimutan. Meron tayo sa Spotify. Diba? Social na tayo. Thank you everyone. And of course, yun nga, makakakuha naman daw ng PDF file. Kontakin namin kayo. Sir, Ma'am Christine, I think pag exit mo yata, automatic siyang magpa-pop up. Again, thank you everyone. See you all next time. Happy lunch. And of course, ingat po tayong lahat and God bless po sa lahat. And of course, in behalf po ng ating Crop Life HRAC team, maraming salamat po for joining. Maraming salamat for the very active participation. Salute po to everyone. Thank you. And see you again sa next webinar or the next activities. Thank you po. Bye-bye po.
Thank you,
Happy
Edward.
lunch.
Thank you, Sir Mo.
Janina.
Thank you.
Thank you. Bye-bye.